Serene Amhishi, and I will be presenting my DNA microarray science fair project. Introduction. The DNA microarray is a tool for figuring out whether a certain person's DNA has gene mutations. A tiny glass plate is housed inside the chip's plastic casing. A few businesses use techniques resembling those employed to create computer microchips to produce microarrays. Thousands of short, synthetic, single-stranded DNA sequences are present on each chip's surface, and when combined, they form the normal gene in question, as well as any human population-specific variations. We did this experiment to determine the effect smoking has on the likeliness of cancer. Abstract. DNA microarray technology has revolutionized the field of molecular biology by allowing researchers to simultaneously analyze the expression levels of thousands of genes in a single experiment. This powerful tool enables the comprehensive study of gene expression patterns, genetic variation, and molecular interactions within a cells and tissues. In a typical DNA microarray experiment, DNA fragments representing known genes or genomic regions are immobilized onto a solid surface and hybridized with fluorescently labeled DNA or RNA samples. The resulting fluorescent signals are then detected and analyzed to quantify gene expression levels or identify genetic mutations. DNA microarrays have a wide range of applications, including gene expression profiling, disease diagnosis, drug discovery, and personalized medicine. Despite its numerous advantages, DNA microarray technology also poses challenges related to data analysis, standardization, and interpretation. Ongoing advancements in microarray design, experimental protocols, and bioinformatics tools continue to enhance the utility and reliability of this versatile technology in biological research and clinical practice. Purpose. The purpose of DNA microarrays is to enable researchers to concurrently examine hundreds of genes' expression levels. Scientists may now examine how genes are activated or inactive in various psychological states or disease states thanks to this technology. DNA microarrays are used to investigate genetic variants, find genes linked to diseases, find patterns in gene expression, and find the molecular pathways that underlie biological processes. DNA microarrays are also used in medication development, environmental monitoring, agricultural research, and personalized treatment. All things considered, the main goal of DNA microarrays is to give scientists an effective instrument for examining gene expression and its part in a range of biological processes. Materials. The materials used are microarray card, microtutor, quick strips of patient cDNA samples, equilibration buffer, hydrazation buffer, control cDNA, micropipetter of 0.5 to 10 microliters, incubator, disposable micropipette tips, handheld UV light, digital camera, safety goggles, and latex or nitrile exam gloves. Procedure. The first thing we did was put on our safety goggles and gloves. We applied 5 microliters of equilibrium buffer with the micropipetter to each spot on the microarray card. To ensure that the samples dry completely, we allowed the microarray to sit at room temperature for 5 minutes. After waiting for 5 minutes, we added 5 microliters of control cDNA to the first well of patient 1 cDNA on the micropipetter plate, and mixed it by pipetting up and down 5 times. We deposited 5 microliters of the combined sample to the appropriate places on the microarray card using the same micropipette tip. For all 4 patients, we did this on every additional cDNA sample. For each well, we made sure to use a new micropipette tip to prevent sample contamination. We again let it sit for 5 minutes, and then when we were done, each spot on the microarray card receives 5 microliters of hybridization buffer, which we then let to sit for 5 minutes. Once dried, we visualized the microarray and observed the results using a long-wave handheld UV light. Analysis. A major trend that's seen in the data is how the patients that tend to smoke for longer periods of time are the ones with a riskier chance of getting cancer. Our prediction was that the 3rd patient would be healthier than the rest of them because he didn't smoke. And it turned out to be accurate. However, we also predicted that since patient 2 quit smoking, she would have a less likely chance to get cancer than the remaining patients, which turned out to be false. Conclusion. The purpose of DNA microarray is to identify any gene mutations present in a specific individual's DNA. We use DNA microarray to figure out what effect smoking has on the likeliness of cancer. We concluded that smoking does indeed have an effect. Smoking less would decrease the chances you have of cancer, although it does not create that much change to their cancer risk factor by itself. Smoking, cancer history in the family, and current major health problems causes huge risks altogether. Thank you for listening.